Welcome students. In this class, we will discuss about good laboratory practice, right? So we are already discussed about good manufacturing practice. We also have good laboratory practice. Let's see what is GLP. GLP is basically an FDA regulation, Food and Drug Authority of America, FDA regulation, and GLP stands for good laboratory practice, and GLP embodies a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, reported, and also achieved. So this is a general definition of good laboratory practice. So GLP embodies a set of principles. It tries to implement a set of principles that provides a framework within which a laboratory studies are planned, then performed, monitored, reported, and also achieved, right? GLP is sometimes confused with standards of laboratory safety, such as wearing protective goggles, wearing of apron, and all other things. So, standards of laboratory safety are different, and good laboratory practice are different. Coming to history of GLP, GLP is formal regulation that was created by FDA, United States Food and Drug Administration, in 1978. Although GLP originated in the United States, it has a worldwide impact. Non-US companies that wanted to do business with the United States or registered their pharmacies in the United States had to comply with US GLP regulations. So not only those companies which are starting, or not only those laboratories which have their uh, services in United States, but also other laboratories present in somewhere else in other countries who want to sell their product in United States, or who want the what can we say the registration in the United States, then they need to comply with US GLP regulations. They eventually started making GLP regulations in their home countries also. So by seeing this, they also started preparing their home countries. In 1981, an organization named OECD, which stands for Organization for Economic Cooperation and Development, produced GLP principles that are international standard, right? So why was GLP created? So we can see in the early 70s, FDA became aware of cases of poor laboratory practices all over the United States. FDA decided to an in-depth investigation and on 40 toxicological laboratories. They discovered a lot of fraudulent activities and a lot of poor laboratory practices. Example of some of these poor laboratory were found practices found were equipment not being calibrated. This is the grave sin, major problem. If equipment is not calibrated, right, properly, then the measurements and all this appropriate things you're taking will go wrong, right? And therefore giving wrong measurements and incorrect and inaccurate accounts of actual lab studies and inadequate testing systems. So these are the issues which they found in 40 toxicological laboratories. For example, they have seen one of the lab that went under such an investigation made headline news also there. So the name of the lab was Industrial Biotest. This was a big lab that ran tests for the big companies such as Procter and Gamble, P&G company. I hope you know a lot of shampoos and other cosmetic products are prepared from that company. So it was discovered that a mice that they had used to test cosmetics such as lotion and deodorants had developed cancer and it died. Industrial Biotest lab threw the dead mice and covered results, deeming the product good for the human consumption. These involved in production, distribution, and sales for the lab eventually served jail time. So, see what they have done. They have tested these lotions to be used on human and deodorants to be used on humans on the experimental animal, that is rat, and that rat developed the cancer and they threw it away. They covered the results and they declared that it is safe to use in human. So this kind of errors can happen if we neglect GLP practices. So GLP is very important to regulate the proper quality products and decorate the laboratory quality. So objectives of the GLP. What are the different objectives of GLP? GLP makes sure that the data submitted are a true reflections of results that are obtained during study. So GLP, if we follow the set of rules and framed by the rules framed by the GLP, it will be sure that the results obtained are reflecting the true results, right? So whatever results you got in the experiments. GLP also makes sure the data is traceable. It makes sure that the data is traceable back, and it also promotes international acceptance of these test results. So changing the test result is the biggest problem with the laboratories. If they're not getting the exact results what they want, they started changing it, and this is not acceptable. And GLP will make sure that all the results obtained during the test reflects the true results, original results. And GLP also makes sure that the data is traceable back, and it also promotes international acceptance of the test. So what are the missions of GLP? So test systems, archiving of records and material, apparatus, material and reagent facilities, then quality assurance program, performance of the study, reporting of the study results, standard operating procedures, and personnel and test facility organization. So these are missions of GLP, right? So first thing, let's start with standard operating procedure. So in whatever laboratory you're working, SOPs are very important. SOP for everything. SOP is a standard set of certain rules for the procedures. It may be for operating of machinery or equipment. It may be for doing any process, any experiment, anything. You need to have certain set of written rules. So written procedures for the laboratory programs is generally termed as standard operating program or standard operating procedures, right? So they define how to carry out protocol specified activities. They are most often written in chronological listing of action steps, step by step. What is the first step? What is the next step, and all? They are written to explain how the procedures are supposed to work. Then what is SOP? It's a routine inspection, cleaning, maintenance, testing, and calibrations. For everything that you should have an SOP. Routine inspection also, how to inspect, what are supposed to be inspected, right? How you will say that after inspection this particular equipment or machinery is qualified, right? Or you are rejecting it, or you will say it is out of calibration, right? So these set of rules. Cleaning, for cleaning also you need cleaning validation, you need rules and regulations, one by one step for cleaning and validation, because you can't clean each and every equipment with the water or with other equipment, other things, right? So cleaning electronic gadgets is in different way, cleaning the laboratory things are in different manner, cleaning the rust forming substances, parts of the instrument you will use a different way. So SOPs for cleaning, SOP for maintenance of the instrument, for the laboratory procedures and other things, SOPs for testing, and even SOP, standard operating procedure, for calibration. So set of rules, right? Chronologically defined rules, step by step is SOP. SOP is mandatory for all these things. These are the actions to be taken in response to equipment failures also, and analytical methods are there, definitions of raw data, keeping records, reporting, storage, mixing, and retrieving the data, right? So all these are very important. Then statistical procedure for data evaluation. That is the next step of the GLP. So statistical procedures are not simply chosen from the textbook. Practitioners in a particular field may adopt certain standards which are deemed acceptable within the field. Regulatory agencies often describe acceptable statistical procedures. Then instrument validation is very, very important. You will have complete validation subject and complete validation chapters in when you come in PG level, master's level. Equipment validation will be there, cleaning validation will be there, process validation will be there, manufacturing validation will be there. So this is the process necessary for any analytical laboratory validation. So data produced by faulty instruments may give appearance of valid data. So the frequency for calibration, revalidation, and testing depends on instrument and extent of its use in the laboratory. Whenever an instrument An instrument's performance is outside the control limits, reports must be discontinued and this particular equipment should be validated. Okay, so instrument validation, equipment record should be included, name of the equipment and manufacturer should be included, model or type of identification should be included, serial number should be included, date, equipment, the date of the equipment, when it was received in the laboratory should be noted, copy of the manufacturer's operating instructions should be there, and reagents and material certifications. The policies, this policy is to ensure that reagents used are specified in the standard operation procedure. Then purchasing and testing should be handled by quality assurance program. Right? So reagents and solutions, unit requirements, reagents and solutions shall be labeled, and then deteriorated or outdated reagents and solutions should not be used and they should be discarded. Included date opened, the date of the opening of this particular equipment should be included, stored under ambient temperature, and expiry date should also be noted. Analytical certification, some acceptable proofs of satisfactory training or competence with the specific laboratory procedures must be established for each analyst. Qualification can come from education, experience, or additional training, but it should be documented properly. Then sufficient people should be there for doing all these things, and requirements for the certification basically varies from certification to certification. Let's see the laboratory certification normally done by the external agencies, like we studied in the last class about NABL accreditation, right? So laboratory certification can also be done by the ISO certification mechanism, right? So the evaluation in concern with issues such as adequate adequate space, ventilation, storage, hygiene. So these are the important parameters which will be considered when certification was given to the laboratories. Specimen and sample tracking is also very important part in the GLP. So it vary among the laboratory to laboratory. They must maintain the unmistakable connection between a set of analytical data and the specimen or samples from which that particular data is obtained. Original source of specimen or the sample must be recorded unmistakably. Okay, so source of the data must be recorded unmistakably and connected with a set of analytical data. Right, documentation and maintenance of the record. This is a very important part of the GLP. Maintenance of the all records, provide documentation which may be required in the events of legal challenges due to the repercussion of decisions based on the original analytical results. General guidelines followed in regulated laboratories is to maintain records for at least 5 years. Length of time over which laboratory records should be maintained will vary with the situation to situation. Important questions to be answered for any analytical instrument. What is the equipment being used for? Is the instrument within specification, and is the documentation to prove this available? I mean, whether this particular instrument is working within the specified limits, and to prove this, any documentation is there or not. If the instrument is not within the specified limits, how much is it deviating? And if the instrument is not within the specification, what action should be taken to overcome this defect? See, these are the important questions, right, before you validate an equipment. Number one, what is this equipment used for? What is its use? Right. Number two, is this instrument working appropriately as per the specified limits? And if it's deviating, how much it's deviating? And it's deviating, what should be done to overcome this defect, to rule out this particular problem? What happens if the workplace does not comply with federal GLP, good laboratory practice standards? What will happen? Let's see that. So disqualification of facility. Before a workplace can experience the consequences of non-compliance, an explanation of disqualification is needed. So FDA states the purpose of disqualification as the exclusion of the testing facilities from completing laboratory studies or starting any new studies due to the not following the standard of compliance set by GLP manual. So possible violation: falsifying information or wrong information for permit registration or any required records or documents, wrong information related to testing, protocols, ingredients, observations, data, and equipment. Failure to prepare, retain, or submit written records required by the law. Then grounds for disqualification. The testing facilities fail to comply with one or more regulations implemented by the GLP manual, they can be disqualified. So the failure to comply, failure to comply to adverse outcomes in the data. In other words, it affects the validity of the studies. Warning or rejection of previous studies have not been adequate to improve the facility complaints. Then consequences of non-compliance. What will happen if you have non-compliance? The FDA states the following consequences of non-compliance. The commissioner will send a written proposal of disqualification to the testing facilities. Then the regulatory hearing on the disqualification will be scheduled. Then if the commissioner finds that after the hearing the facility has complied, then a written statement within an explanation of termination of the disqualification will be sent to the facility. Thus, if it can be shown that such disqualification did not affect the integrity and outcomes of the study itself or did not occur at all, then the study may be reinstated, right, at the will of the commissioner. So these are the consequences of non-compliance of GLP. Upon disqualification of GLP, what will happen? The commissioner finds that the facility was non-compliant on any of the grounds after hearing, then a final order of non-compliance will be sent to the facilities with explanation. If a testing facility has been disqualified, any studies done before or after the disqualification will needed to be determined as essential to a decision acceptable or not. If the study is determined unacceptable, then the facility itself may need to show that the study was not affected by the non-compliance that led to the disqualification. Once finally disqualified, the facility may not or it be considered for a research or marketing permits and study is rejected. So this is what happens when it disqualifies the GLP. What's happening? Students, mute yourself. A lot of disturbance is coming. Yeah. So upon disqualification, what else can happen? The commissioner may notify to the public and all interested persons, including other federal agencies and facilities may have which are in contract. Right. The FDA may ask other agencies to consider whether to support the facility or not under the disqualification. Civil and criminal proceedings may occur at the dissertation of discretion of commissioner. Fines up to $50,000 if one knowingly commits crime, and 1 year imprisonment for registration, applicants, and procedures. Fines up to $5,000 for all others. Civil penalties after failing to improve after a minor violation. Warning was issued. Only those involved in testing will be given civil penalties. Those involved in distribution or sales will be accessed more heavy penalties such as criminal penalties. Getting it? See how many results we will alter and we will change in these things. I hope you know, right? Lot of results are there which we manipulate. But in industries, when you manipulate results, what will happen? What will happen? They can sue you. They can fine you up to $50,000 if you do intentionally, and if you do unintentionally or minor violation, they can fine you up to $5,000, and they can imprison you for 1 year for simply misreporting the data. So be careful. So upon disqualification, the FDA may turn it over to the federal, state, or the local law enforcement. The facility sponsor by terminates or suspends the facility from doing any non-clinical study for permit, and the sponsor is required to notify the FDA in written within 15 working days that the facility is to be suspended. So sponsor is the pharmaceutical company or cosmetic company or the chemical big industry which is sponsoring that research, which is paying for that research, that should immediately stop funding that particular laboratory and immediately should give in written within 15 working days that as per the FDA guidelines, as per the GLP guidelines, so and so laboratory has violated the rules of GLP. So we are discontinuing the funds and we are stopping that particular research. Okay. and reinstatement of a disqualified facility. Once your laboratory is disqualified, how you can restart it back? The testing facility may be reinstated as acceptable non-clinical study to be turned into the FDA if the commissioner can be certain that future studies will be conducted in compliance. So when you make sure and when you prove to the commissioner that next future studies will be done as per the GLP rules, then your permission will be given back. That any current studies integrity have not been severely harmed by the disqualification. And previously, when you have violated the GLP rules, right, it doesn't generally it haven't affected the results of the previous experiment. So that should be informed, and the disqualified facility will be required to put in writing to the commissioner reasons why it should be reinstated and any action the facility will take or have to be taken to ensure any disqualification problem in future. So this is the way how to restart or get the permissions of GLP after once you have been disqualified. Then the commissioner will inspect the facilities. They'll inspect and they'll determine if it shall be reinstated, if it shall be started or not. If it is reinstated, the commissioner is required to notify all the persons that were notified of the disqualification, including the facility itself, that we are giving back the permission because we have inspected the particular facility and they have ensured us that this particular issue or this kind of deviating the GLP rules will not happen in the future. That's why you will send a letter to all the agencies and authorities that the permission is given back to the laboratory. Okay, guys, getting it on? Yes.
Yes, sir.
So this is the short introduction about the good laboratory practice, why it is important, what happened in the past, right, and what happens if you don't comply to the rules, right, and how to get the permission back, right, and the next thing after getting the permission back, and if you do this kind of mistakes again and again, what kind of punishment they will give, all these things we have studied. Yes, finishing this session. So we have seen the definition, right? Already we have seen the definition. Yes or no? So we have also seen the introduction, right? We have also seen the introduction and what GLP stands for, when it is started and everything. Right, we have also seen the history, what happened in the past with the PNG laboratories, how they covered up the results of not showing the rats who have developed cancer and all those things, right? And we have certain set of other guidelines also, like OECD guidelines. We have also seen the current document in the Organization for Economic Cooperation, Organizations for Economic Cooperation and Development, that is OECD principles of GLP. We have seen that OECD principles of GLP was basically revised and updated in 1997. Then WHO has also published standards for good manufacturing practice, covering the manufacturing of drug products and good clinical practice, that is GCP. Right now what we studied is GLP, good laboratory practice. There are also GCP, good clinical practice, and GMP, good manufacturing practice. These three things are also a set of rules and very important. Good laboratory practice are a set of rules to be followed in laboratory. Good clinical practice is a set of rules to be followed in clinics where human beings are treated, covering clinical trials, right, and like phase 1, phase 2, phase 3, phase 4 trials and all. And GMP, good manufacturing practice, is a set of rules which should be followed in manufacturing industries. So these three, good manufacturing, good clinical and good laboratory practice, are very, very important and you should know it. And why should we have it? In general terms, for companies conducting non-clinical safety studies, it is a regulatory requirement, and without this certification or certification of compliance, data will not be accepted by regulatory authorities, because we don't know whether they have manipulated the data, whether it's true or false, and all other kind of things. The emergence of now familiar CROs, clinical research organizations, will become very more popular in the future for non-clinical safety studies. Like we have a lot of clinical research organizations which will do these phase trials, phase 1, phase 2, phase 3, and phase 4 trials, and those results will be accepted by the authorities only if they comply CGMP and CLP practices. Okay, GCP and GLP practices. What is the scope? GLP's requirement in regulatory and non-clinical safety testings of the following test items like pharmaceutical products, pesticide products, cosmetic products, veterinary products, food additives, feed additives, industrial chemicals, and everything wherever they are manufacturing, GLP should be followed in those laboratories. Okay, purpose of GLP principles: to promote the development of quality test data, to obtain reliable and reproducible data, to obtain comparable data between the countries, to achieve international confidence in the study data, to avoid repetitions of studies, and to enable reconstruction of studies, and to optimize the animal conditions and shorten the registration time of the drug. So these are the main purposes of the GLP principles: promoting the development and quality test data, obtaining reliable and reproducible data, obtaining comparable data between the countries, achieving international confidence in the study data, avoiding repetition of the studies, enabling the restriction of the studies, optimizing the animal conditions, and shortening the registration time for the drugs. What are the different elements of the GLP? Test facility management and personnel, quality assurance programs, facilities testing, apparatus, material, reagent testing, test systems, that test and reference items, then standard operation procedures, then performance of the studies, reporting of the study results, storage and retention of records and materials. These are different elements of GMP, right? Test facilities, organization, persons, how all these things are happening, we already see. Test directory responsibilities, what are the directory set of responsibilities should be followed. Principal investigation officer, what are his responsibilities? Basically, he will ensure that the delegated phases of the study are conducted in accordance with the applicable principles of GLP, right? Other details we will see in the next class, like what facilities should be there, how to prevent the contamination, right? How to archive the facilities, how to maintain the documentation, what different documentation should be maintained, right? Test and reference items, how they are maintained and how they are recorded, standard operation procedures, how they are developed, how they are implemented, performance of the studies. All these things we will discuss in coming classes, like reporting of study results and other kind of things. So this is about brief introduction about GLP, good laboratory practice. So following GLP will basically help us in obtaining quality and reliable data, and also remind us to minimize hazardous waste in the laboratories. It is clearly understood that the issue of GLP is obviously so crucial to the modern laboratory operations. The GLP practices have become as an essential ingredient for any professional scientist. So GLP is mandatory to make sure that our data are not fake, to make sure that our data are acceptable internationally, and to build the confidential levels in the regulatory authorities that whatever research we are doing and whatever data we are producing is true and unmanipulated, right? So this is about GLP, good laboratory practice. In next class we will see about the good clinical practices also. Okay guys, that's it for today.